Today we'll discuss a paper that talks about these kinase called Syk and Tau metabolism. Of course, you want to advance, so I'll have to do here. The paper in question is Syk inhibitors reduce tau protein phosphorylation and oligomerization, and we'll go through the experiments that they show to support those claims. In the introduction, the authors tell us a few things that we already knew and some things that we didn't. So, for instance, that tau can be abnormally phosphorylated by various kinases. We had talked about GSK3 beta. We may have had a paper or two that touched on that kinase as well. We talked about CDK5. And then the authors introduced one that we hadn't heard about in the context of this class, but it's a very common kinase. Some people refer to it as the stress MAP kinase because the p38 MAP kinase is an intracellular kinase that gets activated by all kinds of stressors. So any of these have been shown, according to the introduction, have been shown to phosphorylate tau. Then they introduced for us this non-receptor type tyrosine kinase, whose full name is spleen tyrosine kinase, or Syk for short. And they tell us that it has various biological roles, primarily as modulator of immune responses. So it's sort of in the name already. So spleen tyrosine kinase, spleen is an organ that is much involved with blood and blood metabolism. And Syk was identified in that organ, so right off the bat, we know that it has something to do with blood and immune system, right? So Syk has been shown to play very important roles in the context of immunity. Now, we also know that much of what happens during Alzheimer's disease progression has to do with neuroinflammation, and there are certain mechanisms that are very akin to what happens in immune cells when there is an infection. So it's probably not surprising that they may be interested in a molecule that is associated with immunity, because anything associated with immunity could be associated with neuroinflammation and AD. So, in fact, recent studies suggested that there is an association between Syk and AD. For instance, Syk inhibition decreased amyloid beta production and has been suggested to induce the autophagic degradation of tau. And the reason I highlight that in blue there is because this will come back in the paper, the idea of tau being degraded by autophagy. Now, as usual, and we've seen this rhythm in paper after paper, there is some sort of association, sometimes epidemiological, sometimes this I take was individuals injecting Syk inhibitors into animals for other reasons, probably to look at the effect of Syk inhibitors on immune function, and yet finding that they may have had beneficial effects in cognitive function. They may have dissected their brains and found that there was less deposition of amyloid plaques and so forth. So anecdotally, someone makes a connection between a gene and possibly the development of AD, but all of these are correlative studies or studies done in vivo. What we've seen paper after paper is that the authors say, well, we need to look into this in more detail. And in this case, what they ask is what happens when we specifically control Syk inhibition I didn't write it there, but in a neuronal cell line. So outside of the context of immune cells, what happens when you start messing with Syk in neurons? So to do this, they first, and we've seen this before as well, they want to reiterate or sort of reconfirm certain preliminary observations just to make sure that they're starting on a firm ground. And what they do is they check post-mortem samples of people's brains of those who did have AD, those that did have AD or non-AD controls, and then they look for Syk expression within those brains, right? And they see a lot of Syk expression in control individuals and in AD patients as well. They have the, I can't remember what were those two meant to be? They showed two plots, and I can't remember what the difference between those was. But in any case, they seem to be looking at taking a whole bunch of samples. Oh, now I know. It's C1 through C5 and C6 through. So it's just that they're looking at a bunch of samples. They just needed to break it up into two gels. And then they did densitometry analysis on these gels, and they show quantitatively that there is no significant difference in the amounts of Syk protein that you can find in brains of AD patients or controls, healthy controls. Well, not that healthy because these are postmortem samples, but people who didn't experience AD before passing. So my question to you is, knowing that or seeing that there is no difference in the amounts of Syk expression, what would you conclude from this data? Can Syk have anything to do with AD progression? Mm-hmm. Okay, so an idea here, analysts are saying that maybe not, but it is also possible that the changes in Syk levels happen later in life, right? That these differences may become apparent later in life. Josh? The conformation, what is that normally the conformation of a protein will do? Like when you change the conformation of a protein, what are you changing typically? Uh-huh. And the affinities and the substrates and its interactions with others, what do all those describe in a way? Or what do all those... So, if you remember nothing else from college, given your science degrees in somewhat molecular biology, there's this saying, structure, function, right? So you'll hear this over and over again, the relationship between structure and function. That's sort of the whole idea that molecular biology is not magic, that what molecular systems do, they do because there's a structural explanation for proteins and nucleic acids and all the molecules inside cells, right? So structure, function. So yes, once you start inducing via, you said phosphorylation, it could be other things, but yes, I'll take phosphorylation. So via phosphorylation or other chemical modifications, you start altering the structure of Syk. It could be that the levels of Syk are the same, but their function is different. Fabi? Active form and an inactive form. Okay, anything else? Oh, I think I know what you're going. If I paraphrase and make it sound like super, super, super smart, don't say it. But I think what you're saying is super interesting. We tend to, and this is a very natural habit. We tend to say, well, if something is involved in something, if something is necessary for something, in this case, Alzheimer's disease progression, in Alzheimer's patients, I should see more of it, right? But if I understand what you're saying is, well, the levels may be the same, but that doesn't mean that it's not part of the process. So it could be that Syk is still very much part of the Alzheimer's disease progression, even though it may not be its main function, or it may not be related to either its total levels or its activity, right? So this idea that something can be in a system being or playing a necessary role towards progression. So it could be that there's a whole bunch of other things that contribute to Alzheimer's. We know that, but that may not make Syk any less of an important part in the process than that is, regardless of its levels or activity. Okay, so if I understood what you're saying correctly, yeah, the levels could be the same. Maybe even the activity could be the same. And yet, that doesn't mean that it's not an integral part of the process. And the only way to address that question, what would be an experiment that says, yeah, the levels don't change, maybe the activity doesn't change, but Syk is still required for Alzheimer's disease progression? what experiment would you envision? Knock it out, knock it down, inhibit it pharmacologically, do something and show that now these AD patients are not, well, these are dead people, but that you can stop or delay or ameliorate progression of the disease. Cool. So let me leave you guys with a bit of a bitter taste, right? So why measure these things? If you see differences, great. If you don't see the differences, it could be function, it could be necessity, it could be this, it could be that. Well, it's just one piece of information, right? That the levels of protein in these AD patients is not changing compared to controls. So they decide to dig a little deeper because they do believe that Syk has something to do, and they probably think that the answer may be somewhere else, not the levels of Syk protein, but perhaps, like Josh was saying, its activity. So what they do next is look at the co-localization of Syk with phosphorylated tau in AD brains. Remember, they have access to these post-mortem samples. Some of those samples, some of these biopsies, they ground up for Western blotting, like in the previous slide, but some other samples, they subjected to immunohistochemistry. So looking at proteins in tissue samples. And let's first look at what they saw and what they claim, and then have a conversation about that. What they're saying is, when we look at control samples, we see Syk expressed in cells, AD samples express Syk as well, presumably at similar levels as we just saw from the Western blots. When we look at phosphorylated tau using an antibody that detects phosphoserine 199 and phosphoserine 202 in tau protein, we see little to no phosphorylation of tau in control samples. But then when we look at the brains of Alzheimer's disease patients, we see a lot of it. And then if we merge both images, all of the green signal and the red signal coincide and give us yellow signal, indicating that Syk and phosphorylated tau are co-localizing in the same places. So what the authors say in the paper, this is taking almost verbatim from the paper, is that there is co-localization between Syk and phosphorylated tau. Yes, Melita?
Why would they do that certain serine 199 and 202?
So the question is, why look at serine 199 and 202? Those are two of the many residues in tau that have been shown that when phosphorylated, they will induce the oligomerization of tau into these neurofibrillary tangles. So whenever you're looking at tau, having any evidence that it's being phosphorylated in those sites is a good indication that soon, if not already, it's going to be formed into NFTs. I wanted to take a step back because we've seen these co-localization studies before where people look at, you have some green signal, some red signal, and then you look for the yellow signal. We've seen it with all color combinations. And I wanted to take a second here to discuss with you that we need to take these co-localization studies with a grain of salt. And here's why. I'm going to make my argument with these cartoons. And we're going to go back to the images in a second. But let's say that you have these three images from the same cell. Right? So these are not different cells, they're from the same cell. In the first case, you're looking at the green signal. For argument's sake, let's say that you're looking at Syk. Right? In the red fluorescence image, you're looking at the localization of the phosphorylated tau. And then when you do the merge, you see a whole bunch of green dots, a whole bunch of red dots, no yellow dots. Would you say there is co-localization there? There's no overlap, right? We can get into the weeds of this argument of, well, they're co-localized in the sense that they're in the same cells, right? But the argument loses a lot of strength because now they're not in the same places within those cells, right? So we have cells in which you have some green dots, some red dots, and they do not overlap, then the argument of co-localization loses a lot of strength, right? Cool. What happens if the scenario looks something like this? I'm gonna get my steps in today. Now, again, same cell, you see some green dots, you see some red dots, but when you look at the merge, there is nothing but yellow dots. So is there colocalization? Yeah, right? And now we're talking at the subcellular resolution. We're looking at different areas within the cell, and the green fluorescence and the red fluorescence come from the exact same areas within the cell. And I see no green dots or red dots indicating that anywhere where Syk is, you will find phosphorylated tau. This is cartoonish, of course. It's an extreme example, but the concept is the more yellow you see in a case like this, and more importantly, the less green and the less red that you see on by themselves, the stronger the argument there is for colocalization. Now, bringing you back to the image itself, it's kind of hard to tell because we have no other markers, we have no indications of where a cell begins and where a cell ends, but take my word that given the scale that these images were taking, each of those blotches you see on the screen are individual cells. It's not part of a cell. We're not looking inside one or two neurons in each of these squares. We're looking at a tissue with likely hundreds of cells in them, and whenever we see red, the whole cell is red. Whenever we see green, the whole cell is green. Which takes me to this, which is more or less what we see in this image. Would you say that where the green fluorescence comes from all over the cell and the red fluorescence comes from all over the cell, in this case cytoplasm, that when you look at the merge and you see everywhere there is yellow, would you say there is colocalization? Yeah? Do I have anyone for no and an argument for why? What would give you a little pause? Say, well, hold on a second. Are they really colocalizing? Yeah. So we in a way, we discussed this before. These merges, these color combinations are all done after the fact, and it's the computer does for you. It is true, when you're looking at these things, you would normally use lenses that will let the green light only or the red light only. It's not that you look at these cells with your eyes through oculars and you see yellow cells, because the microscope will never let you look at both simultaneously. Fluorescence microscopes will let you look at one fluorescence or another. But then the computer takes the image that it gathered from the green light and from the red light and digitally combines them into a merged image. All right, we're going to follow up on that argument, yeah? In this particular example, you have several cells in the image, right? And each of these green fluorescent or red fluorescent blotches that you see is the whole cell. So if we were to zoom in, so that would be one cell, another one, and so forth. So I'll rephrase slightly what you just said, but it's one of the arguments that it's out there. Sometimes when people encounter these situations in which the entire cytoplasm is green and the entire cytoplasm is red, and then because you see that in the merged image there is yellow throughout the entire cytoplasm, it's a little risky to call it a colocalization. It's because, I'm paraphrasing, these molecules have nowhere else to go. They're all over the place. That doesn't mean that they're necessarily molecularly colocalizing with each other. It's almost like saying, I see green in the cytoplasm, I see red in the cytoplasm, so those things are interacting with absolutely everything there is in the cytoplasm. That's silly. You wouldn't say that Syk and phosphorylated tau interact with every other molecule present in the cells. The only thing that I can think that could do that is water.
So to see if they are interacting with each other, you would have to test it, to knock out want to see. if
Exactly. So it is possible that they're all over the cell and they are interacting with one another. You would have to take an independent approach. You spoke about knocking one out. What other approach have we seen throughout the semester for seeing if things physically interact within cells? Do you guys remember or know from other coursework? What technique do we typically use to say, yeah, protein A interacts physically with protein B? Antibodies. What do you do with those antibodies? So when you say express, I'll change express for present. What I heard something here. Western blotting, but a very special kind of Western blotting. Western blotting will see if a protein is present, but exactly, pull down assays, co-immunoprecipitations, right? And then there are some fancier versions now, and we've seen a couple of them here in class, like you probe in an immunohistochemistry, you bind one of the proteins that you're interested in with an antibody that carries half a fluorescence of sorts, right? And what that means is it carries a molecule that won't fluoresce by itself, but that when in close proximity to its partner, will create a sort of a physical interaction between the fluorophore such that now light is emitted. Those are some of the fancier versions, newer versions of these things. And that allows you to see at physical proximity in tissues. But before that technology was invented, what people would do is, like Jacob was saying, is pull down assays or co-immunoprecipitations. And it fits off of what Ashia was arguing before. You use an antibody to bind to your first protein, you pull it down, and then by Western blotting, you ask the question, is the other one coming down with it?
So the Western blotting is like the expression, like you check.
Regular Western blotting is simply telling you if a protein is present in a sample or not. When you did co-immunoprecipitation, what you did was pull down a different protein, and then with Western blotting, you're asking about the protein that you're interested in to find out if it's interacting with the first or not. So you bring down protein A, and then you use Western blotting to ask, is protein B here as well or not? Right? The answer to that question is yes. It meant that A and B interacted with one another physically. Okay. So once again, the argument goes that when there is staining widespread and somehow, like Josh was arguing, contained within, in this case, a cytoplasm, it's kind of tricky to make the argument that these two things are co-localizing because they're simply occupying the same space. That doesn't mean that they're physically interacting with one another. In the previous examples, when we were looking at punctate stainings, like the one here, the reason why this is a little stronger as an argument is that now these things they could be anywhere in the cell, but they happen to be in the same restricted locations within the cell. So the argument of co-localization gains steam in these cases. The argument here is, well, I see the green dots, I see the red dots, they could have been anywhere in the cell, but what are the chances that they're in the same exact places and very restricted locations if these things are not interacting with one another, right? So if the image was somewhat punctate, that we would have had a different conversation. In this case, I would take the whole argument of co-localization with a slight grain of salt because it could simply be that they exist in the same cells, sure. And when they emit fluorescence and you look at the merged fluorescence, and there's a pseudo image created by the computer, when you look at the merged image, you see that they're in the same spaces, sure. But that doesn't mean that they're physically interacting with each other. So, fine, they took it a step further and obeyed through a weaker argument. They're saying there is no difference in the levels of Syk between AD patients and controls, but we see that in AD patients, Syk and phosphorylated tau interact with one another, co-localize. So we still want to see if the effect of inhibiting Syk has any effect on tau metabolism in culture cells. All of this was in slices of brains from people who had experienced AD before dying or not. So the next experiment they do is take a cell line, a cell culture. This cell line is called M1C, and it's a neuroblastoma cell, meaning it's a cell line made of neural progenitors. These are not fully differentiated neurons. And the fact that it was generated from a neuroblastoma tells you that these cells were coming from a neural cancer, and they had most likely been immortalized in vivo, right? So when you transfer them to a culture, you don't have to really do much more. These cell lines will establish almost by themselves. So what they do first is they look at the effect of known pharmacological inhibitors of Syk activity, and which throughout the paper they call Syk inhibitors. They give us all the details in the paper. These are the drugs and concentrations and everything. But for labeling purposes, they always talk about Syk inhibitor. And they look at two things. First, that these drugs are not going to be killing the cells that they're trying to study. So this is an assay of toxicity. So first, let's make sure that our neuronal cells in culture that overexpress tau do not die when treated with Syk inhibitors. One thing that I forgot to mention is that they purposely use a cell line that overexpresses tau so that they can facilitate the formation of the tangles. When you have a cell that is already, by quote-unquote, design, expressing a lot of tau, you're making that cell more sensitive to any deviations one way or another that will facilitate the formation of the tangles or not, right? If the extreme, the other extreme argument would be to use a cell that has no tau in it, that doesn't express tau. So then there won't be absolutely any point in doing any of the experiments. We know that normal cells are somewhere in between, but by using cells that overexpress tau, they make it a little easier on themselves to be more sensitive to changes in tau metabolism. So anyways, so back to the cells. So these cells that overexpress tau, and when they look at them by microscopy, they don't see any obvious signs of toxicity. The cells are occupying the plate to the same densities that they would in no inhibitor controls. So 0, 0, 1, 1, 10, different doses. 0 obviously is the vehicle alone, so it's either DMSO or ethanol, I can't remember. Now, seeing that a cell is still kind of dividing and still occupying a plate doesn't really tell you much about how healthy they are, so they run two assays. One is called an ATP assay, and it's basically an assay that measures the amount of ATP produced by these cells. And in the x-axis here, you see that there is the various concentrations of the Syk inhibitor. On the y-axis, there is the fraction of ATP produced compared to controls. And for the most part, even though there is that hump there at 1 micromolar, I'm not quite sure why that is. They're not sure either, but none of these differences were significant across the dose curve, indicating that the presence of this drug is not affecting the ability of these cells to produce ATP. And then there is a very famous assay called the MTT assay. The MTT assay is an assay for sort of central basic metabolism. It's basically asking the question, can these cells do glycolysis and oxidative phosphorylation, right? So it looks for metabolites that have to do with regular metabolism, and again, no difference across the board. So subjecting these cells to these drugs is not really doing much to them. So we saw that the brains express Syk, and now we take this cell line that overexpresses tau, we subject them to the Syk inhibitor, and we see that we're not doing anything to these cells by giving them a Syk inhibitor. What silly, or as we would say in technical jargon, trivial reason might explain these results? The fact that you subject these cells to a Syk inhibitor and they don't seem to care. If you were a devil's advocate, what could you say? Well, but... Exactly. The trivialist of reasons to explain this is, well, maybe these cells don't express any Syk. You saw Syk expressed in the brain, but the brain has all kinds of cells, including many that are of pseudo-immune origin, like microglia, astrocytes, right? So what if all that Syk that we saw in the brains comes from those glial cells and not the neurons? Maybe these neuronal precursor cells don't express any Syk. So, absolutely right. So what they did next was check that these cells do express Syk. And what they find is they do express it. So you see M1C, 1, 2, 3, and 4. Those are different clones of these cells, but essentially the same parental cell line. And there is expression of Syk across the board. As a control, they use these HEC291T cells. That's a cell that is completely unrelated to neuronal precursors or anything like that. And when they normalize the expression of Syk to the expression of a housekeeping gene, in this case GAPDH, they see that the expression on M1C cells is less than in this HEC291T cell line, but who cares? There is plenty Syk there, right? So the reason that these cells were not sensitive to Syk inhibition was not as simple, it's not as trivial as to say, well, they don't have any Syk. They do. All right. So now that they have cells that express a lot of tau and that have Syk, they will inhibit Syk. And the first thing they see is that when they treat the cells with these Syk inhibitors, there is a significant reduction in the amount of tau. Now, tau-5 is an antibody that will detect the totality of tau protein, no matter what state it's in, right? And what these plots are saying is that as you increase the dose of the Syk inhibitor, there is less and less total tau present. They use GAPDH once again for as a housekeeping for loading control. And when they do the densitometry and do all of the necessary corrections of total tau levels based or relative to total GAPDH levels, they come up with these numbers. And it turns out that with increasing doses of Syk inhibitor, there is less and less tau. I said that NI was no inhibitor before. Now looking at this law, I realized that I made a mistake. NI means not induced. And it is taking these cells that I said overexpress tau, but inducing them to overexpress tau or not. So not induced would be the cells before the induction, when they have normal levels of tau or very little levels of tau. But then you induce them, and then they express all of those gigantic levels of tau. And when you expose them to Syk inhibition, they will express less and less and less. So apologies about that. So NI is not no inhibitor, is not induced. It's the cells not being forced to express lots of tau. So you see my question down there for everybody. You see this significant decrease in the amount of tau protein present in these cells. What could lead to this downregulation of a protein following an experimental treatment? How can you explain less protein in a sample following a manipulation? Being degraded? What else?
Broken down.
Degraded.
Broken down, potato, it's about the same.
Maybe like during the treatment, mRNA is being produced to a certain extent, and then it starts to eventually be translated.
So if you see protein, you see less and less protein with the treatment, and you're inclined to blame it on the mRNA, what would you say? That what happens to that mRNA?
Really need to make as much mRNA with
Okay.
less protein.
So less mRNA.
Yeah, less than mRNA.
There you go.
Yeah.
Clara?
Oh, maybe,
Yeah.
like maybe changed forms, like you can't detect it.
Okay, interesting. So it's not that it's not there, it's that for some reason it changed form enough that the antibody can't detect it anymore. Okay, interesting. Any other ideas? Removed from the cells. Okay. Less translation. All of these are totally valid reasons, right? So all of these count. And typically what you would do is you would try, depending on your level of interest in what the ultimate reason is, but typically you would try and go through all of them. So for less translation, you would do one of these chase experiments in which you stop translation, then you see how long the protein lasts. That would be for degradation as well. For export, we would measure the levels of tau in the supernatant, maybe in the space around the cells. But the one approach they took, which allegedly is one of the easier ones, is go for maybe the differences in protein are reflective of differences in mRNAs. Maybe the inhibition of Syk is doing something to the transcription or the stability of the mRNAs for tau. So let's look at tau mRNAs, and they did. And when they looked, they saw no difference. Right, so this decrease in the protein levels could not be explained by decrease or concomitant decrease in tau mRNA. Must have been explained by something else, either different translation rates or equal translation rates but increased degradation rates or export rates, or an idea that I hadn't originally contemplated, that it's still there, it's just that we can't see it because it underwent some sort of conformational change that makes it invisible to the tau-5 antibody. That's an interesting option too. Okay, so now that observation that the mRNA levels were the same led the authors to the next question, or one of the next possibilities, and that is maybe it is being produced at the same rate because the mRNA is still the same, and they probably assumed that the translation rates were comparable, but they want to explore the possibility that maybe it is being degraded more. And if you guys remember in the beginning, the authors in the introduction emphasized the fact that tau can be degraded by autophagy. Autophagy is one of the mechanisms that cells have. Many years ago, it was thought to be something really serious, like the cell was chewing itself up from within because it's so sick and starving and desperate for resources that it would destroy its own molecules to survive. Nowadays, we know that that is still true in many cases. Autophagy is related to things like starvation, energy depletion, and so forth. But we also know that it happens at a decent baseline rate as simply a mechanism for recycling. Okay? So anytime that certain molecules are degraded by autophagy, that's simply a mechanism to recycle components, and maybe in a way it's the cell asking itself, if I needed what I just destroyed, if I still needed it, I'll make it again. But what if I didn't need it anymore? Maybe I can use these building blocks to build something else, right? So the authors decide to look at autophagy, and what they do is they treat the cells, the M1C cells, with the Syk inhibitors as usual, and they look at two things that are closely related to autophagy. And I have a cartoon here that I stole from the internet to illustrate the point. Whenever there are proteins that are either misfolded or that are targeted for autophagic degradation, they are labeled. You know how in the past we've talked about ubiquitinylation of proteins? For autophagy, the label that marks proteins for the autophagic pathway is called P62. It's a small protein that is attached to things that need to be recycled and basically tell the cell, we need to recycle this. Right? So the machinery for autophagy, these autophagosomes, is built based on bits of membrane that come together and form these autophagic vesicles within which the molecules targeted for autophagy are degraded, right? And in the formation of those vesicles, of the autophagosomes, a key component is this LC32 complex that is really the joining of LC31 and another protein called PE. So when you're trying to understand if cells are doing more autophagy or less autophagy, the approach that you can take is you look at LC32, or the ratio between LC32 and 1. That gives you an idea of how many of these autophagosomes are being built. The more LC32 that you can see in a sample, the more of these autophagosomes were being built. And you look at the levels of P62. And you may be thinking, well, if P62 is important for autophagy, then I should see those levels increase as well. May seem a little counterintuitive, but the more autophagy there is in cells, the less P62 you will see. Why? Because P62 gets degraded within those autophagosomes as well. Okay? So an increase in LC3, the LC32 to 1 ratio, as shown here, based on Western blotting, and a decrease in P62, both significant, is an indication that those cells, in the presence of the inhibitors, are making more autophagosomes, and they're degrading more of that P62. So it's a good sign that there is more autophagy in those cells. And even though they don't show us any additional evidence, they use this as an argument to explain why it is that those cells with the Syk inhibitors have so much less tau protein. Even though they have plenty of tau mRNA, they may have a lot less tau protein because the protein is being made, but it's also being degraded by the autophagosomes. All right. Next, they decide to look at not so much what Syk inhibitors do to tau levels, but to tau phosphorylation. Going back to your question before, the phosphorylation of tau in various residues is what induces the formation of these fibrillary tangles. So as tau gets phosphorylated in certain places, it will acquire a series of conformational changes that will make it more prone to forming the fibrillary tangles, making the neurons sick, right? So what they do is they look at phosphorylation of tau using phospho-specific antibodies. So here on the screen, you'll see that each of the blots is labeled with some name, some acronym, PHF1, CP13, AT180, and so forth. Those are the sort of the common names of these antibodies. In red font, what I did was indicate what specific phosphorylated amino acids they recognize. So PHF1 will recognize tau that was phosphorylated in serine 396 and serine 404, CP13, serine 202, and so forth. Okay? And in all cases, they see that there's this drastic reduction in the phosphorylation of tau in response to the Syk inhibitors. Right? So drastic reduction in all forms of phosphorylation in response to the inhibitors. They do the densitometry, and they find that when the cells are not induced to express a lot of tau, there is little phosphorylation. Of course, there's very little to phosphorylate to start with. But as the cells are induced with no drugs, a lot of phosphorylation. But as you add the drug, you start seeing, so no induction down here, induction but no drug, lots of phosphorylation. And as you add the drug, that phosphorylation decreases. So what they say is treating M1c cells with Syk inhibitors led to a significant decrease in phosphorylated tau. And that statement is not incorrect. There is less phosphorylated tau inside of these cells. But if you guys remember, there was also a significant decrease in total tau protein caused by Syk inhibition. Could the drop in phospho-tau levels just reflect that there is less tau in cells to phosphorylate? So what we're doing here is we're getting really picky in that it's one thing to say there is less phosphorylated tau. Those are the facts. We can see that. But it's a totally different story to say there is less tau phosphorylation. That would indicate that given the same amount of tau within a cell, if Syk is active, there's going to be plenty of it phosphorylated. If Syk is inactive, there will be less phosphorylation of tau. It's a different story. Because the Syk inhibitors are causing a drop in total tau protein, it may be Just as simple as that. The less phosphorylated tau is reflective of the less tau there is altogether. Can we really say anything about the process of tau phosphorylation based on these results? The answer is short. The short answer is no. We need to do some sort of normalization. And they did. So what they did is they calculated, they also measure total tau in these samples, even though they don't show us the plots, they show us the densitometries in the paper. And when they do the ratio of phosphorylated tau in any given specific amino acid to total tau, they see that there is a significant drop no matter what the phosphorylated site. And they also use immunohistochemistry to show that there is many cases in which you can see total tau, which is shown here in red, and you see less phosphorylated tau, which is shown in green. These, I know there's a lot of images, but you can think of them as 4 blocks, right? So the first block is for PHF1, second for CP13, AT180, and AT270, right? And in all cases, the top plot is the control. You see many cells with tau and many cells with phosphorylated tau. When you treat them with the drugs, here they give us the name of the drug, there is plenty cells that have tau and fewer cells that have phosphorylated tau in them, right? So with both, what they're saying is that not only are the levels of phosphorylated tau decreased, the phosphorylation rate of whatever tau is left is less as well. It's like a two-pronged thing. The levels of tau are decreasing, and on top of that, whatever is little is left is not being phosphorylated as much. So this leads to the idea that Syk would normally lead to the phosphorylation of tau molecules, right? And how do we phosphorylate tau inside of cells? Do you guys remember? Some of the molecules that we mentioned at the beginning? Ooh, that's the phospholipase. How do we phosphorylate tau? There were those 3 that I mentioned, right? The kinases, and which ones? Do you remember from the intro? GSK3 beta, CDK5, and P38, right? For some reason, they skip CDK5. They look at GSK3 beta and P38. Now, here we need to take a step back and mention quickly, we had seen this before, so this should be sort of a refresher. It was a refresher for me. GSK3 beta is tricky. Typically with kinases, phosphorylation activates them. GSK3 beta is a bit of a special case in which phosphorylation actually inhibits it, right? So the running hypothesis here is that somehow, and I'm going to annotate this on the slide, somehow Syk is inducing the phosphorylation of tau. And the hypothesis is that it is by activating GSK3 beta and activating P38, right? So that's sort of what they claim. So if that is true, then when you inhibit Syk, you should see that both GSK3 beta and P38 are less active. So how do you make GSK3 beta less active? By phosphorylating it. And that's what they see in their samples. In their samples, they see that when they add the inhibitor, GSK3 beta is more phosphorylated than before, right? So there's more phosphorylated GSK3 beta than before adding the inhibitor. With P38 is more classical in the sense that it gets activated by phosphorylation. So what they see is that when they add the inhibitor, that one there means 1 micromolar of the inhibitor, there is less phosphorylated P38. And to the right, there is the total levels of both proteins, just to show you that the lesser signal, or in the case of GSK3 beta, the more signal of the phosphorylated forms cannot be explained by changes in the total levels. That is really an increase in the relative amounts of the phosphorylated, or a relative decrease in the phosphorylated forms. Okay? So this checks. Syk inhibition leads to deactivated tau kinases. It will lead to the phosphorylation, therefore deactivation of GSK3 beta, and phosphorylation, less phosphorylation of P38, therefore less activation. Then, Josh, you brought up PP2A, right, or PP2. PP2 is the phosphatase that normally takes out phosphates from tau. So another way to regulate phosphorylation of tau is to take its phosphates less from it, right? So you can increase the levels of phosphorylated tau by simply dephosphorylating tau less. So here what they're going to investigate is whether PP2 might be deactivated by Syk under normal conditions, and therefore if treating with Syk inhibitors might turn on PP2, right? So how do they look at activation of PP2? They make use of something that we hadn't really discussed about PP2 before, but I illustrated in this cartoon that I stole from the internet as well. It turns out that PP2 can exist, like so many other things, in two states, active or inactive, right? And it will be active when it binds to a magnesium ion in its catalytic site, and that is mediated by the methylation of a specific residue or a specific amino acid in PP2. So often we've seen that phosphorylations are the things that turn on or off proteins. We just saw it for the kinases. In this case, it's a methylation that will allow PP2 to bind the magnesium ion and be active. But there is this thing called PME1. It's a demethylase. When PME1 gets its hands on PP2, methylated PP2, and takes the methyl group out, now the enzyme can no longer hold on to that magnesium and it cannot perform its function. So it acquires an inactive state. All of this to say that demethylation of PP2 means inactivation. Okay? So what they saw is they looked at the methylated PP2 levels in cells, right? In other words, when they see that the levels of methylated PP2A drop in response to the inhibitors, that means that PP2 was becoming less active with the Syk inhibitors. Does that make sense? And if with the Syk inhibitors, the PP2 is becoming less active, that's an indication that PP2 will be taking phosphates away less. This is a little counterintuitive now that I present it, because if it is true that the methylated levels drop, that means that there is less active, more inactive, so then you would expect that phosphates are taking out less from tau. So this is a little counterintuitive. They say that treating M1C cells with Syk inhibitor led to an activation of the kinases and a drop in demethylated PP2A, which means an accumulation of the inactive form. No, sorry, of the active form. Dimethylated, no. I'm so sorry about that. I'm confusing myself, but I'm now realizing that it was confusing in the paper to start with. So they say that Syk inhibitors led to activation of PP2A, but I think that they got this wrong, because methylation of P2A is associated with activity. Let's put a pin on that. Don't worry, there won't be any questions about this next week, because that's confusing. All right. And in the minute or two left, looking at tau phosphorylation, that's nice. But ultimately, what do we care about tau in the context of neurons, whether tau oligomerizes and forms these fibrillary tangles or not, right? So how about tau oligomerizing? And what they do is they conducted some Western blots. Up there it says minus BME. BME stands for beta-mercaptoethanol. Beta-mercaptoethanol is a very strongly reducing agent that when people do Western blots, they use to make sure that whatever weird, funny conformation a protein had in space becomes linearized, and this beta-mercaptoethanol, what it will do is break those disulfide bridges that typically tend to keep proteins covalently attached to one another, right? So it's a way of really keeping all of the peptides that were present in a protein separate from each other. But if you take it out, now there will be peptides that are covalently bound to one another. That's what happens when tau oligomerizes. When tau oligomerizes, now these peptides come together, and whenever two cysteines come in close proximity, they can form a disulfide bridge. Without beta-mercaptoethanol, you're not going to break those bridges, and they will remain attached covalently to one another, which means that they're going to become two peptides become twice the size, right? And 3 and 4 and 5 becomes 3, 4, 5 times the size of the original peptides because they're all covalently attached to one another. So in a Western blot, what that does is it forms these comets in a way. I know Jacob brought up the comets last week, but these forms that are moving a lot more slowly because they're piling up on one another and becoming bigger and bigger molecules, right? So what happens is when they're not looking, when they have gels without beta-mercapto and they're looking at cells without the drug, there is a lot of oligomerization of the tau peptides. When they add the drug, there is a lot less of the oligomeric forms. And if they look with the phosphorylated antibodies, same thing. A lot of phosphorylated peptides oligomerize. If you add the drug, there is a lot less. They quantify, and they call these tau high molecular weight, phosphorylated tau high molecular weight, and there is a lot more high molecular weight tau in controls than in cells with inhibited Syk. But then we go back to this idea that look at the monomeric form. The monomeric form is also dropping drastically. So when they do the regular adjustments, they see that when you look at monomeric form and then you do the ratio between high molecular weight and the monomeric, high molecular weight and the monomeric, there is no significant difference. Right? There's one slide extra here because they were not convinced by this data, so they decide to look by immunohistochemistry, and they use an antibody that detects only oligomeric tau. And what they claim is that when using that antibody, there is a significant drop. So the evidence when it comes to oligomerization is a little conflicting, right? You look at one approach, no change. You look at a different approach, and there seems to be a drop. All in all, their conclusion is shown in this cartoon. I don't want to take any more of your time, but essentially they're saying that Syk normally keeps the levels of tau low through autophagy, high by inhibiting autophagy, keeps the levels of GSK3 beta and P3 MAP kinase high, and that it normally inhibits PP2A. When you inhibit Syk, autophagy will go up, meaning the total levels of tau will drop. The activity of these kinases will drop, so phosphorylated tau will drop as well, and the activity of PP2A will increase, so there's going to be less phosphorylated tau, though we need to take a closer look into that because I don't think they got that.